Experiment number 3. In this experiment we're going to count the frequency of each of the 64 codons in chromosome 1 of human DNA. The aim of this experiment is to show that there are multiple mathematical balances between the frequencies of different codons. So we need to start the software by clicking here. It takes about 10 minutes for the software to loop through the whole of chromosome 1, so I'm going to cut short the video the software has finished looping through chromosome 1, and what we do now is we copy, Ctrl+C, all of these frequencies and paste them into Excel. This is the genetic code table. The frequencies are all laid out according to where the codon appears in the genetic code table. The frequencies in the blue section, the top half of the table, sum to the same number as the frequencies in the bottom half. So the sum of the blue section is shown here, some of the green section is shown here, and the ratio of the blue to the green is 1.000696. Similarly, if we take the left and right sides of the genetic code table, the frequencies of occurrence of the codons in the blue area sum to the same number as the frequencies of occurrence in the green area. The ratio of blue to green being 1.001. If we take the first row and last row of the genetic code table, then the sum of the frequencies of the codons in the first and last row is equal to the sum of the frequencies of the codons in the middle two rows. The ratio of blue to green is 1.001. If we take the first and last columns of the genetic code table, then the sum of the frequencies of the codons in the first and last columns is equal to the sum of the codon frequencies within the middle two columns, and the ratio of blue to green is 1.002. If we take the four corners of the genetic code table, the sum of the frequencies for these four corners is equal to the sum of the frequencies for the central four cells here, and the ratio of blue to green is 1.003. So as you can see, there are multiple symmetries within the genetic code, and so our next step is to find out what is causing these symmetries.